Assalamualaikum warahmatullahi wabarakatuh. Thank you for coming to this presentation. I'm glad to see you all. Let us introduce ourselves. My name is Tiara Sesilia. My partners is RINA AZZAHRA, RITA ALAWIYAH, RONA APRILIA UTAMI, and YOLANDA PUSPA SARI. In this presentation, we will discuss about antimicrobial effect of various combination of plant extract. First, Yolanda will explain to you about introduction. Then, Rita and Rina will explain to you about materials and methods. Finally, me and Rona will explain to you about result, discussion, and conclusion.
Introduction. Naturally derived compound and other natural products may have application in controlling pathogens in food. Delacroix and Maza, 1995, Bowles and Juneja, 1998. Some spices are known to contain essential oils that possess antimicrobial activity, such as eugenol in cloves, allicin in garlic, and cinnamic aldehyde, and eugenol in cinnamon. In addition, some vegetables and herbs contain substances that inhibit microbial growth. Chinese chef Allium tubersum lodler belongs to the same family as garlic, onion, and leek, and is an important ingredient in Asian cooking. The goal of this research was to search for a combined extract with effective antimicrobial activity and better flavor for use in various food products. Material and methods. 1. Edible plants. Cinnamon and cornifacture were obtained from a traditional Chinese pharmacy at Neipu, Pingtung County, Taiwan. Chinese chef was purchased from a local market at Neipu.
2. Microbial strains. 8 species of bacteria. Basilicus subtilis, Escherichia coli, Flavobacterium sp, Listeria monocytogenes, Pseudomonas aeruginosa, Salmonella typhimurium, Staphylococcus aureus, and Vibrio parahemolyticus, and 2 species of molds, Aspergillus flavus and A. niger, were obtained from stock culture at Department of Food Science and Technology, National Pingtung University of Science and Technology, Neipu, Taiwan. 3. Preparation of extract. Cinnamon or corny fructose in parentheses 10 grams was extracted with an equal amount of 95% ethanol in parentheses weight or volume in a water bath at 508 degrees Celsius for 1 hour in parentheses U 1982. After filtration through Whitman #1 filter paper, the residue was re-extracted with addition of an equal same amount of ethanol and then filtered. The combined filtrate was then concentrated and a rotary evaporator. 4. Preparation of test microorganisms. Bacteria were grown on plant corn agar in parentheses PCA or in broth in parentheses PCB, pH 7 and 30 degrees Celsius, except for Pseudomonas aeruginosa, which was ground, and Luria bertani, medium and V. Parahemolyticus, which was ground, and Bouillon, medium. PDB, pH 7 at 30 degrees Celsius.
5. Antimicrobial tests. The antimicrobial tests were carried out according to this diffusion test. Sterile filter paper was impregnated with 10 milliliters of various combinations of plant extracts and placed in the center of the agar plate on which the test microorganism was uniformly inoculated. 6. Application input system. Fresh part was selected, ground, added with the cabinet extract, and used to make dumplings with 0.01 and 0.2% potassium sorbate added as contrast, and deionized water as the blank. The dumpling samples were stored in the refrigerator at 48 degrees Celsius. Commercial fresh guava juice was added with the cabinet extract with deionized waters as the blank. 7. Statistical analysis. The typical data were subjected to an analysis of variance for a completely random design using statistical analysis system programs. The Duncan-New multiple range test was used to compare the difference among means at the level of 0, 05.
Result 1. Synergistic effect of various combinations of plant extract. Initially, 105 species of plants, vegetables, and Chinese medicinal herbs were studied separately for their antimicrobial activities. 3 plants were then selected for their excellent antimicrobial properties. Individually, each extract of cornifructus, cinnamon, and Chinese chive showed strong inhibitory effect on bacteria, on yeast, and on molds respectively. Result 2. Temperature effect. Heat treatment, commonly used in food processing, are boiling 100 Celsius and autoclaving 121 Celsius. To study its thermal stability, the combined extract was processed at 60, 80, 100, or 121 Celsius for 0, 5, 10, 15, 20 min and used for antimicrobial tests, and the results were shown in Fig. Result 3. pH effect. The pH of processed foods usually range from acid to neutral. The combined extract with the original pH of 3,4 was adjusted to pH 3, 4, 5, 6, 7, or 8 with 6 and HCl or 4,5 and NaOH solution and stored at 48 Celsius for 24 hours.
Number 4. Metal ion effect. To stimulate the aqueous environment in various food products, the combination extract added with each metal ion to yield a final concentration of 1 or 10 and used for antimicrobial tests, and the results were shown in the table 3. Number 5. Food addictive effect. To maintain or upgrade food product with desired quality, food-grade chemical additives were commonly added during food processing. Therefore, the combined extract was added with commercial food additives to yield a final concentration of 0.10% or 0.1% and used for antimicrobial tests. And finally, conclusion. The combined extract of cornifructus, cinnamon, and Chinese chive were used to evaluate its antimicrobial activity on common foodborne microorganisms, including bacteria, yeast, and molds. Thank you for listening. May those we have shared be beneficial for all of us. See you. Wassalamualaikum warahmatullahi wabarakatuh.